Good morning, everybody. I hope you can hear me. Hope everyone's had a nice weekend, and we can get started today on choline. Before we do, I just wanted to go over a few things because I'm sure you're all curious about the test next week. So, with respect to the test, first and foremost, I just need to reiterate this. I won't expect you to memorize things like the chemical and metabolic pathways. So memorizing those things, one, it's pointless because they're available to you. It's like memorizing the periodic table. Two, I don't think that's actually going to test your knowledge of anything. I do want you to understand the general message related to them, and there may be some key little pathways. Not necessarily memorizing them, but you need to understand them, and we go through them, so it's not just related to the table. It's why we're talking about them. But again, I don't expect you to memorize that kind of thing. With all vitamins and minerals in general in this course, you should understand digestion, how it's transported in the body, their main primary functions and mechanisms, and what deficiencies and toxicities could look like, as well as some information related to how and why these types of deficiencies and toxicities are dealt with in Canada specifically. So we generally want to focus here. So things like think of fortification, dietary recommendations, restrictions, things like that. Now, with respect to the test, it's going to be on weeks 1, 2, and 3. So we're not going to cover this week because I don't think it would be fair for me to go over this week and then you have a test a few days later. So it's just going to be in the first, second, and third week. The midterm itself is going to be online. So that means that I do have to make it open book, which is fine. It's going to be multiple choice, and it's based on the material that we've covered in slides and lectures, as well as required readings, not the recommended optional ones. So the required readings is fair game. The recommended optional ones, I won't be testing you on. And again, I don't expect you to memorize specific things about the readings. I need you to just understand the take-home messages, maybe the results, what they found, what they didn't find, things like that. And given that with respect to vitamins and minerals, realistically, we're just primarily like folate's the big one. You know, it's obviously your final exam is going to be more that's backloaded. Though it is multiple choice and it's online, you shouldn't take it lightly. Because it's open book, because you're going to be presented with one question at a time and it's timed. So you have 50 minutes. It's during this exact class time next week online. And after you answer the questions are going to be locked and you won't be able to go back and change them. So the answers will be shuffled. So what that means is that you do need to know things. You can't just expect to have your notes out and everything. And the answers will be shuffled, so no two tests are the same. Now, that being said, there is actually going to be a review meeting. I don't know how many of you saw the announcement. It'll be hosted by our two amazing TAs in this course. The session is going to take place this Wednesday, February 2nd from 3 p.m. to 4 p.m. And the Zoom link can be found in the Quarkus announcement. So I believe it was either Katie or Mike that made the announcement. I am not sure. You will have to check with Katie and Mike on that. They're fully responsible with respect to it being recorded. I'm not totally sure. So you'll have to talk to them and ask that. You might be able to actually write that in the discussion. Or I can ask them. But either way, you can ask them maybe even at the beginning of the session. Somebody who's there can ask if it's going to be recorded or not. Generally, I don't think so. I wouldn't count on it, but yes, there is a lecture this Wednesday. So there's the lecture on B12. It's continuing this. Are there any more questions? Amy?
Sorry if you already mentioned it. Can we go backwards on the test?
No. So that's a good question. So that's what I meant by when I said it's locked. So once the questions are locked, my understanding is that I think you've already had experience with that in other classes. I know it's a pain. I wish there was sort of a better system to do it, but I mean, the final exam, the plan is to have it in person, so you'll have everything available. And again, good news is that we're back in person the Wednesday after that, so you can actually, once you write the test and everything, you can tell me in person how much you hated it. So that's always great. I made the test a little bit earlier this semester too, because I know you have other work this semester with this course. So I didn't want to inundate you with tons of materials all at once. So that's why normally this week I would have included, but again, I tried to stagger it as best I could. So yes, and just to reiterate again, this course, I cleared it with admin for the remainder of the semester. Once we go back to in person, we'll have Zoom sessions on Mondays still. So we're not meeting in person on Mondays, because Monday mornings for a 50-minute lecture, I don't think is worth anybody coming in to sit in a lecture hall. But Wednesdays will be in person. There are going to be 45 questions on the test. I have a few more questions that I need to add on there, but there will be 45 questions on the test in 50 minutes. Okay, so for somebody that just asked that, I should probably read the questions in the chat first. Okay, so that being said, we're going to today talk about choline. So this week is choline and B12. And again, not going to be on the test, but please pay attention. This is pretty fun stuff. Let's dig into it. So choline is, as we know, or we don't know, the most well-known part of choline is the polar head of phospholipids. So choline's attached right here. This is the phospholipid head. Choline's attached to a phosphate group, which is esterified to glycerol and two fatty acids to form phosphatidylcholine, which is the most common phospholipid, and it makes up about 50% of our cell membranes. Now, it's not technically a vitamin, since we do make some, but we also need to get choline from food to have enough. So an adequate intake and upper limit levels were established for choline in 1998. So again, a little bit more relatively recent, although 1998 doesn't seem that recent now. Choline's required for methyl group metabolism, and we'll go over the metabolic pathway that you don't need to fully memorize, you just need to understand. It's also involved in neurotransmitter synthesis and the structural integrity signaling functioning of cell membranes. It's part of that phospholipid bilayer. One of these neurotransmitters, unsurprisingly, as the name implies, is acetylcholine, which, as I hope some of you know by this point, is very important for muscle contraction as well as neural functioning and development. So choline is made by the liver, but evidence does suggest that it's not enough. However, it's found in many foods and mostly animal products, unsurprisingly, because of the way it stores in us as mammals. It's found in other mammals and other animals outside of mammals, as you'll see. There are some plant sources as well, but mainly red meat, poultry, fish, dairy, eggs, and then you have peanuts, soy, and wheat germ, and they have choline in them. Half of our food sources come in the form of phosphatidylcholine, which is, again, it's in cell membranes. So that's unsurprising because it's in the cell membranes, and a lot of the things we eat, most are composed of cells. Now, you may have seen this ingredient before, lecithin. Lecithin is actually a phosphatidylcholine-rich fraction, which is prepared during commercial purification of phospholipids. And it's a term that's often used interchangeably with phosphatidylcholine. It's used as an emulsifier in foods that have both water and fat. So think of things like chocolate and margarine. So those things would be things that are, when they're commercially prepared, they would use this kind of an emulsifier. So you'll see that in those ingredients. Choline's also in breast milk, so most infant formula also adds it. They add it into the vitamin mix of formula to help the product match breast milk composition. Now, it's not typically found in prenatal supplements in Canada, but there are some that are available. And as you can see here, there's some of the sources where you can get it. As you'll see a little bit in a few slides, if you have 3 ounces of delicious beef liver, you'll get most of the choline you need for the day. But these are the foods that are fairly rich in choline. So there's good plant sources, which include soybeans, potato, wheat germ, kidney beans, and quinoa. Now, in terms of absorption, our gut bacteria could impact the bioavailability by breaking down choline to betaine prior to it being absorbed. Betaine's a derivative of choline, and it's made in the liver and kidneys by removal of a methyl group. Betaine is also called trimethylglycine. And less choline is available for absorption if a bunch of the bacteria have actually metabolized the betaine already. Betaine is also found in foods. Now, betaine itself does have an important role in the body, but we need choline as well. So even though you might have a lot of like foods that have betaine in them, which some of the foods are actually similar to choline, you may not be getting enough choline still. And as we'll get to as well, choline deficiency is not generally a big problem. In foods, choline is found as both water soluble, so free choline, phosphocholine, glycerylphosphocholline, and lipid soluble forms, so phosphatidylcholine and sphingomyelin. There is a lot of $10 words here I have to deal with this morning that are really challenging my ability to keep up with coffee. Free choline is absorbed in the jejunum and the ileum through a carrier or passive diffusion. So I'll get to the next slide. You'll see that really briefly. And the pancreatic and mucosal phospholipases actually break down some phospholipids in food. So choline is taken up right here. Choline is taken up by the intestinal cells by transporter through facilitated diffusion, and it enters the portal circulation. Now, phosphatidylcholine or sphingomyelin enters the lymph through via the chylomicrons. And that's really, we're not, as you've seen with folate, we're not going to get into the super nitty gritty of it. I don't expect you to. That's pretty much as exciting as it gets with that. Now, most of the processing is done by our liver. We can make phosphatidylcholine, that's PC, from other phospholipids like phosphatidylethynolamine, as well as we have choline, which is catalyzed by phosphatidylethynolamine and methyltransferase, that's PEMT, and it uses S-adenosyl methionine, SAM, as a methyl donor. You may recall SAM from the previous lecture. With folate, we use most of the choline we consume and endogenously and make it endogenously for phosphatidylcholine. So phosphatidylcholine can be used to make sphingomyelin, which is part of myelin. So as you know, with myelin and cell story conduction through the Schwann cells with nerve functioning, it's very important. So we need myelin; it's critical. If we aren't using choline for phosphatidylcholine, it's converted to betaine, which again, as I just mentioned, is important since it can donate a methyl to homocysteine to generate methionine via betaine homocysteine methyltransferase. Now, as you can see here, let me see, sorry, I put the cursor in the wrong spot. If you'll notice here, there's no B12 because B12 is not a coenzyme for this. So I'll follow the bouncing ball on this. So the other thing is if we're not using the choline can actually join acetyl coenzyme A in the brain to form the neurotransmitter acetylcholine. So again, really important for brain function. And this is just to show you the sort of interplay between methionine, folate, and B12, which we'll be talking about on Wednesday. As you can see, choline does play a role here with respect to methylation and the entire cycle here. Can't see the bottom of my screen, but that's okay. So the major roles of choline are in formation of acetylcholine and, again, phospholipids for cell membrane signaling. Phosphatidylcholine is also part of very low density lipoproteins and low density lipoproteins, which, as you know, some of you may have learned, move fat from the liver to the tissues. Now, choline deficiency can result in increased fat in the liver, which is not great. And as you'll see a little bit later, is actually a way we measure whether we have choline deficiency. But it can also be potentially negative if choline's too high because because if VLDL and LDL are raised, as we know, these are typically referred to as bad cholesterol. Now, again, in nutrition, I always like to reiterate there's no good or bad necessarily. It's very gray, but as you can see here with choline, there's a bit of a balancing act with respect to that. So these VLDL and LDL, as we know, are associated with cardiovascular disease. How much of a major role they play in it depends, I think, on a lot of other factors. But regardless, choline choline deficiency and excess amount can result in some sort in possible negative effects on the health. And we may get too high choline again, as we've learned in this class, and we'll be repeatedly learning through supplementation. So over supplementing with certain things. Choline can provide methyl donors and methylation, as we've seen, of homocysteine to methionine. The enhanced methyl donor supply and DNA methylation status has actually been suggested to be associated with neural tube defects, along with the role of phosphatidylcholine in producing sphingomyelin. So in addition, the same mechanisms might be involved in other birth defects, such as the orofacial malformations, as we know, with cleft palate. And again, these are associative. So these aren't, again, with folate, we have a better idea of sort of the relationship with these things. Cell membrane structures as part of phospholipids, which are part of cell membranes, influence the conformation and function of membrane-bound receptor channels. So phospholipids also affect cell function by serving as second messengers and as precursors for prostaglandins, and all that to say that it is important for cells. I can let you go through this slide. Basically, the take-home message is phospholipids make up about the 30% of lipid weight of the milk fat globule membranes. That's the MFGMs. And sphingomyelin, phosphatidylcholine, and phosphatidylethylamine make up 30% each of the total population content of MFGMs. So in that plays a role with how we develop formula as well as breast milk. So this kind of all informs that. Now, maternal choline is necessary to make phosphatidylcholine to give to the fetus to incorporate into the tissues. Phosphatidylethylamine and methyltransferase is low or absent in placental and fetal tissue. So this is why it's important for the mother to make sure that the fetus gets it. Choline can help methylate if it's needed, which would occur in a B12 deficiency. And then there would be less choline available for phosphatidylcholine and sphingomyelin production, which could negatively impact the fetus. So it's essentially, it's like a prioritization. So if there's a B12 deficiency, it kind of shifts gears with respect to where the methylation is actually needed. But we also need choline too. So this is why healthy, balanced diet, making sure that the mother is getting enough nutrients for both herself and the fetus. In terms of storage, our cell membranes are the largest storage pool with limited amounts in the liver and kidneys. And cells are, as we know, everywhere. So in terms of nutritional assessment and common approaches, because choline wasn't always considered an essential nutrient, it's not always tested in foods. And so the nutrient databases aren't always complete for choline. Plasma choline can be measured, and it reflects recent dietary intake. And even if someone has low choline levels in the body, the plasma level is never less than around 50% of normal. And this is why a deficiency is not super common. And you can also measure metabolites in the blood, betaine and dimethylglycine. And low choline is associated with liver dysfunction. So another thing that we do to assess it is test liver function via alanine aminotransferase. Now, again, just something to note with choline that's important is that one of the things that we do is we measure it through food frequency questionnaires and 24-hour recalls, as we know with diets, and how we measure people's dietary patterns. And one of the problems is that we don't have enough information on choline, and that doesn't just say that like we have to do an adequate intake level for choline only. That also it tells us that some of the foods that people are logging into the 24-hour recalls or the food frequency questionnaires, we don't have the information. So we don't necessarily know if there's choline in that food or not. So let's say people are reporting this, the amount of choline that they're taking in during the day, and you'll see a slide in a couple of slides about people's reported choline intake, and that informs us of the amount that they have. But the problem is some of the foods that they're inputting may actually have choline, and we're just not able to capture that because we don't have that kind of information. So there's definitely lots of room for research here. Now, the primary criterion, as I said, used to estimate choline is the adequate intake, and the primary criterion that we use to measure adequate intake is the prevention of liver damage as assessed by measuring serum alanine aminotransferase levels. So the adequate intake for adults is 550 milligrams per day of choline for men and 425 milligrams per day for women. Now, these values were based on the amount of 7 milligrams per kilogram per day, which was the amount that was found to prevent hepatic alteration in men. And that, again, is defined as elevated levels of alanine aminotransferase concentration in the serum, and a reference body weights of 76 kilograms and 59 kilograms for men and women, respectively. Now, there's no nationally representative estimates of the intake of choline from food or food supplements. So there's definitely gaps in information on choline here. And choline in the diet is available as free choline, or it's bound as esters, as we discussed, phosphocholine, glycerophosphocholline, sphingomyelin, or phosphatidylcholine. Now, phosphatidylethynolamine and methyltransferase activities actually increased by estrogen. So premenopausal women are more resistant to a low choline diet. So the AI for women is lower than men as a consequence of this. Now, the demand for choline increases during pregnancy and lactation. Choline is transferred from the mother to the child via placenta, as we saw, and through breast milk. And as we saw, this is why formula actually tries to match the breast milk. And similar to B12 and folate, the adequate intake for infants is based on breast milk. And for older children, it's extrapolated from adults. So in terms of its relationship with liver enzymes, the adequate intake for adults was determined from this one study alone and was only on men, and they only studied the 2 doses of 500 milligrams per day and 13 milligrams per day of choline. So pretty big difference. And again, as you can see, there's definitely a lot of gaps here with respect to the kind of the information that we have from choline. Now, the adequate intakes were set based on preventing fatty liver disease and liver damage seen in the deficiency that we talk about. It's rare in humans, but it has been documented in choline deficient clients and people getting TPN without choline, so getting parenteral nutrition without choline. Studies like that are choline deficient. Studies like that show choline deficiency they found the potential effects on the liver. But again, in terms of trying to sort of put this into the general population, we obviously have quite a few gaps, which is part of the reason why we only have adequate intakes for choline in this case. Now, what's interesting in terms of the upper limit for choline that we've developed is that the intake levels are higher than that would typically be consumed from food. So that's how we define the upper level. So the critical adverse effect from high intake of choline is actually hypotension. So blood pressure just drops. Blood pressure was super low at about 7,500 milligrams of choline per day, which is a lot. Think about how many 3-ounce beef livers you'd have to eat for that amount of choline. So the upper level for adults was set by dividing that actual dose by an uncertainty factor of 2. This is something that's just sort of done, and saying that it's 3,750, then rounding down, because that was a super high upper limit. So you don't want to set it at 7,500 because there's so much uncertainty between 7,500 and the adequate intake that we set. So they want to just kind of play it on the safe side. Now, other potential effects, hence the image, are fishy odor, sweating, and liver damage. Now, fishy odor is a pretty fun one. You can Google up choline, but excess amount of choline does result in you actually emitting a fish-like scent from your body odor, as I imagine Aquaman would. Fishy odor is a result of trimethylamine, so trimethylamine is formed in the actual gut bacteria. So when you have excess choline, the bacteria form trimethylamine from the unabsorbed choline, and as a consequence, you end up having this scent. It's an unpleasant scent. Not super common, obviously, but yeah, some people might just work around fish regularly and they don't have issues with choline either. So. Now, comparing intakes to adequate intake will overestimate prevalence of inadequacy. So the EAR is used for inadequacy estimates, but choline doesn't have an EAR. So we'd have to use other criteria to determine a likely deficiency. So there's some American data, which is lower than the AI, but there's no actual national Canadian data. Again, gaps in knowledge. Data from smaller Canadian studies in pregnant women did find intakes less than AI. So I have these papers posted. So Masick found 306 milligrams per day. And then the Lewis study did a 24-hour recall and found that the mean total choline intake value during pregnancy was about 347 milligrams per day, with about 23% of participants meeting the adequate intake recommendation. During lactation, the mean total choline intake value was 346, with 10% of participants meeting the AI recommendation. And eating eggs made a big difference. Now, again, as we discussed, not all foods have necessarily been tested for choline. So it's possible that these mean intake levels that we're getting, and remember that these don't seem to meet the adequate intake, any of them really, depending on who they study, the population, but like with men, no. With women, same thing. None of these seem to meet the adequate intake. A lot of these used, as you can see, food frequency questionnaires, 48-hour recalls, and so that doesn't inform us fully that they're not getting enough choline. It may well be that they are getting enough choline, and we just don't have the information on the foods that they've input to indicate whether there's enough choline or not. So, again, super quick lecture today. Let's turn to talking about active areas of research. Okay, so infant neurodevelopment's first one. We know that the demand for choline increases during pregnancy and lactation, and it's due to increases in a 1-carbon metabolism. Now, choline's added to some infant formulas, and research has found increased maternal choline leads to increased breast milk choline. So it's been suggested that choline is important in the neurological development of the unborn child, and low maternal choline intake during pregnancy can alter DNA methylation in the placenta and cord blood. Now, there's an inverse relationship between the risk of neural tube defects and maternal choline intake or plasma choline concentrations, and that's independent of dietary folate or supplementary folic acid. Now, inverse relationships, as we've learned with the lectures on looking at scientific research, help inform us, but part of the issue is that there may be another factor involved that's resulting in low choline intake. And so it could just be that they're just not generally eating enough too. And as a consequence, they're not getting enough choline. So again, this is where we have gaps in literature. In terms of animal studies, they do suggest an actual causal relationship, unlike those maternal studies. And the causal relationships between cognitive function and dietary availability of choline during development. But this hasn't been established in humans. So some studies exploring choline and cognition in humans are ongoing, but we don't have enough evidence or results to indicate that. So if you're really interested in cognition and choline, I suggest that maybe you pursue that once you finish your undergrad and go into grad school, because we do need more information on it. And again, with this idea of choline being involved with acetylcholine and cognitive development and whatnot, we have been thinking about also memory loss with aging and what role it could possibly play, as well as potentially even an effect on Alzheimer's. But again, I would say that we want to look at memory loss first and then Alzheimer's and dementia after, depending on what population we're looking at. But it's been studied in stroke patients and patients in cognitive defects because, as we know, as the brain ages, there's a decrease in phospholipids in neuronal membranes, leading to decreased structural integrity, which again, potentially supplementing with choline could mitigate that. Choline might also have a positive effect on cognitive function later in life, since it serves as a precursor to acetylcholine. Choline's been used as a treatment for actually used as a treatment for stroke, and that's under the hypothesis that increasing blood levels of choline through supplementation could actually increase the availability of phosphatidylcholine, which in turn could have a positive effect on membrane structure and potentially result in improved memory function. Now, we've seen in Alzheimer's patients that choline levels have been found to be lower in them. But a Cochrane review of 12 randomized controlled trials concluded that there's no evidence for the use of lecithin in dementia. So they looked at lecithin, you remember that in the first few slides. Review paper also did note that due to the wide variation in study time and the type of choline sources used, it wouldn't be possible to conduct a meta-analysis yet on the topic. All that to say, there really isn't enough information and we need more. And I think this is actually a pretty important area of research in terms of the effect, the influence and impact of nutrition on Alzheimer's and dementia and later on in life. So other active areas of research, the non-alcoholic fatty liver disease. So as we've talked about, choline deficiency can induce abnormal phospholipid synthesis that might lead to the development of non-alcoholic fatty liver disease. Evidence of this does come mainly from animal studies or select patient populations. So again, people on total parenteral nutrition without choline who develop steatohepatitis that's actually reversed by the addition of choline. But the question is whether choline could actually prevent non-alcoholic fatty liver disease to the general population hasn't been established. So we don't know that yet. And again, there's a lot of factors too. So if we increase our choline intake, maybe it'll prevent it. But is that going to make a difference if our alcohol intake is too high, which we know can have a severe impact on on fatty liver disease in general, not non-alcoholic fatty liver disease. So there's a lot of things to consider with respect to that. Like, will it mitigate that? Will drinking and choline mitigate it potentially? We don't know. None of these are all questions we're not sure of. And I hope I'm stimulating some of you to maybe look into this later on. I'm trying to encourage people to go into research. In terms of cardiovascular disease, it's another question. So there's some research, but it's not conclusive, and it's possibly related to the reduction of homocysteine levels by choline. But other evidence suggests that higher choline intake can lead more gut bacterial use of choline, yielding the metabolite trimethylamine and oxide, which has been associated with increased risk of cardiovascular disease events, possibly due to increased atherosclerosis. Now, again, as we know with atherosclerosis, there's a lot of other factors that could potentially affect it. So this could be one factor. So this may impact cardiovascular disease to some extent. Or it may not make a difference because there's other circumstances that are having more of an impact on CVD, such as other dietary patterns or lifestyle patterns. So there is some concern that choline may affect lipid metabolism unfavorably, as we've discussed with VLDL and LDL and the relationship there. So on one hand, the presence of choline in the form of phosphatidylcholine is essential for cholesterol and triglycerides to be transported by very low density lipoproteins from the liver to the vessels. But on the other hand, we also have the beta-ine homocysteine methyltransferase enzyme, which can increase secretion of very low density lipoproteins. And beta-ine intake has been found to be associated with LDL cholesterol levels and lower HDL cholesterol levels. So not only is it increasing LDL levels, it's decreasing HDL levels, which could help shutter some of that. So again, we're not 100% sure. And again, that's all under the assumption that it's exclusively this discordance of of blood lipids that's affecting our risk of cardiovascular disease and atherosclerosis. And as we've learned with the lecture on science and what of impact other things outside of cholesterol might have on atherosclerosis, we're not completely sure if it'll make a big difference. In addition, there's also genes that are related to PMT and the activity related to PMT, so they can contribute to inter-individual differences in choline requirement. So again, with the AI, we have like the shotgun approach of providing this requirement, but some people might need a lot less, some people might need a lot more. And as we know, if some people require a lot less choline because of the differences in that PMT activity, and they're getting too much even if they had adequate intake. So this is part of the difficulty with sort of this approach of giving general recommendations. They're generally pretty good, especially with some other nutrients and vitamins, but they might not be that great with other ones where we have less information, choline being a great example of that. Now, the differences that we're seeing in choline requirement, the differences in this choline requirement because of this mutation as well, could also contribute to the inconsistency that we see among studies. So we do need to consider that in future studies. And we need to recruit in future studies. We need to recruit more people of different ages, definitely looking more at the effect on women, because like a lot of research, we don't have enough information on women. So it's a lot of these things that we need to start considering for this kind of work to look at later on and see the effect. Now, the systematic reviews in general say that there's some observational evidence that choline during pregnancy might be beneficial for the neurological health of the child. In adults, choline may have beneficial effects on cognition, but high quality intervention studies are lacking. Results in the effects of choline on body composition, blood lipids, and cardiovascular health were inconsistent. So, I'll throw this question to you to just think about, because it's, just basically, should choline be included in prenatal supplements? And I think that's an important question to sort of think about with this. But that's the lecture for today. That's choline. It's a short one because I go into B12 right after this, and that's going to be on Wednesday's lecture. Are there any questions related to this that I can hopefully answer? Slide 10, take home message for slide 10. Yes. The take home message here is that phospholipids make about 30% of total lipid weight of the milk fat globule membranes. So that's really one of the important messages here. And that it could potentially be important for cognitive development. Does that make sense? Okay, good. You're welcome. And again, if you don't have questions right now, you want to go over the slides, you can always post in the discussion, and I will do my best to get back to it quickly. The function of choline. That was sort of the whole lecture, was the function of choline. In terms of the function of choline, we're looking at development of the big things are acetylcholine, developing acetylcholine. Let's go back to this. I'll just go on the slide so I'm not sort of. Yeah. So major roles, formation of acetylcholine and phospholipids for cell membrane signaling, as well as being phosphatidylcholine is an important part of VLDL and LDL. So, in general, I just think of this as choline, because I know there's different derivatives of choline in this case, but it's also part of VLDL and LDL, which move fat from the liver to the tissues. So just to reiterate this, deficiency can result in increased fat in the liver, and it can also potentially be negative if choline is too high, which could happen from supplements if VLDL and LDL are raised. So if choline's too high, it could raise VLDL and LDL, which, as we know, is implicated in cardiovascular disease. If choline's deficient, it can result in increased fat deposition in the liver, which is how we've measured choline deficiency. Jason, you had a question.
Yeah, hi, Professor. I just have a question about slide 14. When you were talking about the AI and the metrics. You mentioned a lot of stats that are not included in this slide, such as that the 7 milligrams per kilogram, 7 milligrams of choline per kilogram was the metric used to determine AI for men. Are details like that going to be testable on the midterm?
I see, for this midterm, no. In terms of choline, we're not looking at choline in the midterm. So none of this is going to be on the midterm, just for specifically choline and B12 on Wednesday, just because I don't think it's fair to test you guys when I'm just throwing this material at you now. But in terms of details like that, I'm going to, like specific RDA, I don't like testing you on memorizing certain numbers with RDAs and RDIs. I want you to understand the physiology. I want you to understand the functions, why these things are important. So it's more about understanding it. In the nitty gritty details, if I was going to give you a multiple choice test where you could go back with an open book, I'd probably test you on details like that, just because it's fair game and you have time to go back and whatnot. But in this case, I don't think that would be a fair thing to test on memorizing that. But in terms, so just to reiterate, again, we only have, like you said, the value in men of 7 milligrams per kilogram per day, and then they made that assumption with women as well, just because it's standardized to body weight. But as we also know with women, estrogen, the fact that they have estrogen in their body, or higher amounts of estrogen in their body, because men produce it as well, also indicates that they may not require as much choline, so that number could be lower. So we're just sort of extrapolating that data based on very limited research.
Okay. To elaborate on that, the digestion section that was covered in week 1, should we put a little time studying the pathways that were all mentioned? For example, like bile production and stuff like that.
I would put a little bit of time into it just so you understand the general synopsis of it. If you're looking at metabolic pathways and there's components that we've highlighted a few times, then those things I look at. But if you're looking at, like, let's say there's random pathways I'm thinking of, like glycolysis, where ATP is donated or things like that, I don't want you to, I don't care for you to memorize every little component of the pathway. I think it's a waste of your time. Just sort of understand why we're studying this pathway, what the outcome of the pathway is.
Okay. All right. Thank you, Professor.
No problem. Let's see. Matthew had a question. Components of cell membranes that choline is part of. You had mentioned something about prostaglandins. Do you remember what slide that was, Matthew? I'm trying to figure out what you're asking. Let's see here. It was the last section of the function slide. Well, okay, so I'm trying to kind of figure out what. So in terms of choline, with respect to choline, they're part of phospholipids, which are part of cell membranes. So that's really the take home here, and that they influence the formation of receptors and ion channels. So they're part of that important barrier with the cell. They also affect, phospholipids also affect cell functions by serving as second messengers. So the phospholipids themselves, and as precursors for prostaglandins. I think that's what you were asking. But again, it's just all of this to say that it's critical with cell function. You're welcome. Trying to go back in the chat here, sorry. Materials from week 2 going to be on the term test? Yes, week 2 materials are going to be on the term test. It's a scientific method and whatnot. Julia, you asked, after February 7th, will there be both in-person and recorded lectures? Yes. So at the beginning of the class, I was just talking about that briefly. So Monday mornings, this lecture, we're going to be continuing to do it online. Because it's a 50-minute lecture, and Monday mornings, I know people probably don't all love Monday mornings. But Wednesdays will be in person because it's the longer lecture, the hour 50-minute one or 2-hour one. For the active areas of research, what should we remember? I don't necessarily want you remembering or memorizing, but I do want you to understand active research and how it works. So, in terms of the scientific method and active research, you should understand that. Given that we only have 3 weeks of material on this midterm, there may be a decent amount that's on the scientific method. What I would suggest is attending the tutorial that Matt and Katie are putting on, because they will go through quite a bit of questions pertaining to that. Because right now we're a little short on time for that, and that's why we're having the tutorial session. So I had a question. If the recorded lectures will be posted, and the answer is they're not posted. I don't actually have recordings for all of them either, because these are supposed to be live in-person lectures. I don't generally post those ones. So if it was an asynchronous class, then yeah, of course I'd post them. But this class has a scheduled time, so I won't be posting them now. Okay, everybody. So if you have any further questions, please ask in the discussion. You could always email me too. And that's it for everything today. I hope you've enjoyed the interesting gaps in knowledge in choline, as well as the things that we do know. And I hope everybody has a fantastic day and a great week. Take care. See you Wednesday.